Today you can see the research retreat for cancer at UCL. Behind me there's a huge hub of activity as young scientists interact with senior professors to discuss all aspects of cancer. Cancer at UCL spans basic science through to clinical practice and clinical trials that transform cancer care. It's a fabulous and vibrant environment, and all people here are enjoying the day.
My name is Karina Burger, or Kate, and I'm working in Sergio Quesada's lab and Carl Pack's lab. I'm a PhD student in the Cancer Institute in my third year now. So I'm working on immunotherapy of melanoma, specifically on CD4 T cells that have cytotoxic potential. Now that is not sort of the normal phenotype you would think that they would have, because normally the CD8 cells are the cells that are cytotoxic and active in an immune response that way. And CD4s were more of a regulator and helper phenotype. However, the cells that I am using, CD4 T cells, from the TRP1 model, are cytotoxic. Now the TRP1 model is a model where you have a mouse that is completely deleted of this one gene, TRP1, and this is a melanoma antigen. Now they're also knockout mouse for RAG, that means they usually wouldn't have any lymphocytes, and they additionally have a knock-in of a TCR that is specific against this melanoma antigen, TRP1. Now, because of the forced expression of this, you do get CD4 T cells. So effectively, the cells that I'm taking out of this model are CD4 T cells that are reactive against a melanoma antigen presented in class 2. Now, that is really important because these cells, when you inject them into a tumor-bearing mouse, they directly go into the tumor and directly target the tumor cells and kill them very efficiently. Sergio has already published this, and he found that in this setting, which is lymphodepletion and anti-CTLA-4, together with adoptive cellular transfer of these cells, at the end, when we dissect the lymph node and the tumor, we find that they're cytotoxic and that these cells are actually also the reason why the tumor is eradicated. They also perform long-term protection later when you re-challenge them. They protect the mice forever from melanoma, effectively. And I'm trying to find out how they do it. Why are they cytotoxic? How are they producing still all the cytokines that are needed for the helper phenotype, but why do they still express granzyme B and are that cytotoxic and eradicate the tumor? And effectively, I'm just looking at different genes and trying to figure out and pinpoint which ones are very important for that.
My name is Pedro Velissa. I'm a postdoc and I work with Ron John Chakravarty and Hans Staf at the Royal Free campus up in Hampstead. The idea of this project is to explore one particular signaling pathway, which is called mTOR, in engineered T cells. So we want to further engineer cells that are engineered to kill tumor cells. And so what we've shown here is that by manipulating this pathway, we can either make the cells better killers, but then they die quicker, or we can make them worse killers, but they stay more of them stay in your body. So it's a two-edged sword, and hopefully we'll be able to take the best of it. We're working at the Royal Free Campus, so we're physically a bit removed from the rest of the Cancer Institute, but that hasn't been a problem because we come to seminars here, we keep in contact, we share reagents. I know pretty much everyone here, so it's a good environment. People collaborate.
I'm Lydia Lee. I'm a clinician, so I'm a hematologist. And I'm working at the Cancer Institute with Kui Yong and Martin Poulet in the Department of Hematology. I'm looking at the immune system, and the immune system has really potent machinery. We've got the ability to kill cells, and we know that because we can kill bacteria when we have infections, for example. However, what we want to do is redirect that machinery towards cancer cells. So by definition, cancer comes about because it's evaded those mechanisms. And what we're trying to do is redirect the immune system to be able to fight cancer cells using your own immune system. I got interested firstly by the clinical side. So because I'm a hematologist, I wanted to work in myeloma, and UCL has a large dynamic myeloma department. So I approached them because I knew they were doing some interesting work. Cancer Institute is a great place to work because it's large, it's dynamic, loads of interesting things going on, and it's great to be a part of it.
My name is Lucy Young. I'm a PhD student and I'm in Pablo Rodriguez-Veciano's laboratory. So what we've tried to do with this project is to describe how a molecule called SHOC2 forms a complex with protein phosphatase 1 and one of the RAS family GTPases called MRAS. And what we've shown is how this complex interacts with a molecule called SCRIB and how together they compete for the binding of the phosphatase. And with the result of that is that they have contrasting functions within the pathway. So depending on where in the complex the PP1 is found, this pathway can be negatively or positively regulated. And what's interesting about this is that we found that it was involved in migration and sort of polarization of cells.
Hello, I'm Jonathan Lederman. I'm Professor of Medical Oncology at the Cancer Institute. I've been here since the beginning of the Cancer Institute, and I head one of the research departments, the Cancer Research UK and UCL Cancer Trials Centre, very much involved with phase 3, phase 2 trials, taking work from the Cancer Institute and from other clinicians within the hospital into multi-centre trials throughout the UK and abroad. More recently, I've become the clinical director of the Cancer Institute, and my role is very much to help to bring together the work that's going on, in particular in translational research, with the clinical work that goes on in UCL hospitals, which is a very big cancer centre. We're really quite uniquely placed at the Cancer Institute, not only being part of a multi-faculty university and funded by Cancer Research UK in one of their Cancer Research UK centres, but we are also attached to a very large clinical cancer centre of UCL hospitals, which is really well known nationally and internationally. So we've got the opportunity through the patients that we treat and through the scientists in the Cancer Institute of really developing innovative therapies for these patients. So my new role as clinical director in the Cancer Institute is very much to bring these two parties together and to plan the future for both clinicians and scientists in the Cancer Institute. So this is the 6th annual conference of the Cancer Institute. It's been a very successful day and as previous annual meetings have been also, because it brings together the scientists, the clinical scientists and the clinicians, because it's very much a conference that spans both the basic science, the translational work, and, as we'll hear later this afternoon, some of the clinical activity that's going on within the hospital and the Sarah Cannon Research Institute, which is part of the connections that we have through the UCL Cancer Institute.